Thank you for joining us for the seventh seminar in the Plant Phenome Journal monthly webinar series. My name is Carolyn Lawrence Dill. I'm an associate professor at Iowa State University and an associate editor at the Plant Phenome Journal, who handles our speakers article, and I'll be moderating this discussion and participating next month, May 7th, as Dr. April A.G. Carroll, one of the people currently online, discusses the North American Plant Phenotyping Network that she chairs and I co-chair. You can watch past webinars from the TPPJ on our new YouTube channel. Just search for the Plant Phenome Journal or at the TPPJ website, where you can read about cutting-edge work going on in plant phenomics. So our speaker today is Dr. James Schnable. He's an assistant professor at the University of Nebraska-Lincoln. He was also the winner just last week of the Rhodes Early Career Maize Genetics Award at the 2018 Maize Genetics Conference, which was in Saint-Malo, France. So if you missed that, you really missed out. Today, James will be speaking about his recently accepted TPPJ paper and associated work titled Comparing Plants Measured on Non-Overlapping Time Points Using Functional Data Analysis. With that, I'm going to turn off my video and hand it over to James.
All right. Thank you very much for that introduction, Carolyn, and thank you to the Plant Journal, both for accepting our paper and for this opportunity to speak. Now, on my title slide, I have a number of people lined up here, Yumo and Yuhang, who are both statisticians and co-authors, and Yuhang was the lead author on this paper, as well as some other collaborators here at UNL who are contributed to various projects I'm going to talk about. And I'll try and mention these people again as I go through the slides and again highlight them at the end of my talk, because I think one of the things that really sets plant phenotyping apart from a lot of other fields is you need expertise in such diverse areas. I mean, it is not something that really any one lab has the relevant expertise to do. So I'm fortunate to be in a network of such great collaborators, and I want to make sure that I give them the credit that they deserve for both their expertise and general helpfulness. So actually, I think all of the data in this talk, with possibly one exception, comes from a greenhouse phenotyping system at the University of Nebraska-Lincoln. This is a very carefully drawn diagram, as you can see, where we have 4 different cameras, and we have plants in rows in the greenhouse, and the plants in the greenhouse can come out and be photographed with each of these 4 cameras, which I won't go into in any particular detail today. The important things to know about the system is that if you look at North America and you look at the public sector, the facility at UNL cannot grow the most plants. It can't grow plants at the lowest price per plant. The comparative advantage that we have here is that we can grow really big plants. So we can grow corn or sorghum up to about 2.3 meters, which allows us to look at reproductive stage development. So that's our comparative advantage at UNL. That's where we focus a lot of our work. So here you can see some actual pictures of the phenotyping system we work with. We're able to grow about 672 plants at any given time. And as I said, we can grow these plants large enough that we can look at adult stage maize and sorghum. When we do this, we get a lot of different types of sensor data, image data. What you see here are data from 4 of the 5 different types of cameras. An RGB camera that's essentially the same as a really nice DSLR, photographing the plant from different angles, and then some other cameras like a hyperspectral fluorescence and thermal infrared. Excuse me. So when we talk about plant phenotyping, it's important to break this down. There are really 3 different separate domains of expertise, and I always like to include this introduction because depending on who you talk to, plant phenotyping can mean different things. So the first of these is what I would consider the engineering challenge, which is once we can grow some plants and we'd like to collect some pictures or other data from those plants. The second challenge is more of a computer science or machine learning problem, which is that once you have pictures like the ones I showed you on the previous slide, you have to turn that into some measurement of something you care about from the plant in order to actually gain biological insight. So this is our computer vision machine learning challenge. And then finally, once you have a bunch of numerical measurements of the plant, you have to be able to figure out what to do with those numbers to get back to either identifying genes controlling traits, making predictions about what plants will perform in what way in what environments, all the interesting biology. So this is both a statistics and a quantitative genetics challenge. So so we have, we do this imaging, we can take photos of plants, we see the growth of the plant over time. This is really good for outreach because, I found actually a really good way to engage either school children, farmers, it's a different way to look at plants. This challenge of how do we go from these pictures to lots of measurements of the plant, I think is probably one of the biggest hurdles we face, but it's not the one I'm going to talk about today. Instead, I'm going to focus on a couple of very simple traits that we already know very well how to measure from image data, things like height and biomass. Because that lets me focus on a statistical challenge and how improving statistical methods can help us to address some real limitations of the engineering challenge of how we actually collect phenotyping data. Now, I showed you this picture before, but one important thing to know about the phenotyping facility at UNL is it was originally designed to only grow 200 plants, and each plant was going to be imaged every day. We then extended the facility so we could grow more plants, which made me very happy, because with 200 plants, it's very hard to do quantitative genetics. The problem is that, let's see, I guess my mouse sort of shows up on the screen. While it's easy to add more plants, it's very difficult to increase the throughput of the actual data collection. And I think this is something you would generally see in a wide range of different types of plant phenotyping. Adding plants is relatively cheap, but adding more capacity to collect image or other trait data can be quite expensive. This isn't just the hardware. For example, with drone-based imaging, you have to actually have someone who can fly the drone, and you have to get them to the field. And anyway, there are lots of these bottlenecks. So this created a problem. For a field experiment, or really any quantitative genetics experiment, even 672 plots, so each plot plants, is a relatively small field experiment. Here in the greenhouse, we can grow 672 plants, but we can only image about 400 of them together on a single day, which most conventional methods for being able to look at traits and identify genes really do require that we have data from all the plants at the same time. Now, this is not a unique problem to UNL or to plant phenotyping, and so there are a number of strategies people have come up with to deal with the idea that if you don't have data from all the plants and all the days. Perhaps the most basic traditional approach is just to drop the data points. I have a grad student right now. He's working on a very different project and was working with a really large GWAS data set and from many, many years and many, many locations. He decided to do this. And so he went from about 300 genotypes and 60 traits to about 10 traits and 50 genotypes. And we had to walk through the fact that this was not going to work for GWAS, and we needed a solution other than just dropping lines and data sets with lots of missing data. Another option is just to fill in average values. Again, there are some obvious problems with that. Perhaps the most relevant one for this conversation are what are called parametric models. So for example, if you look at plant growth is often modeled as a sigmoidal or logistic curve, where at the beginning you have a very small plant, the plant grows really slowly, the growth accelerates, and then slows down as you reach flowering maturity. And a sigmoidal or logistic curve like this can be defined really by only 3 parameters, which are highlighted here. This was a very last-minute slide. Anyway, one is the sort of peak value that you reach, so the difference between your starting point and your end point. Another is essentially the rate of growth, so what is the time from when you start growing to the point of maximum inflection in the curve, and the final is that slope of that curve. So with just 3 values, you can model this growth curve, and then you can try and take all of your data points and fit them on this growth curve, and project the data points that are missing for any given sample. And this actually works reasonably well, at least for some traits like biomass, most of the time. The problem is that as a biologist and my training was as a geneticist, not as a breeder. The times when models don't work are the stuff that we're really excited about. So it's a bit depressing if in order to work with these types of data we have to throw out all the really interesting and weird exceptions. And the solution to this, and this is something I didn't even know about until I started collaborating with the statisticians here at UNL, is to use what are called non-parametric models. So while a parametric model can be defined by a small number of variables in a function, non-parametric models don't require prior assumptions about what shape of curve you're going to see in the data, but just try and fit whatever shape or pattern is there, and should allow us to capture interesting weirdness than parametric models. And this was the focus of the paper that was accepted to Plant Phenome. So about the same time we were realizing we had this real problem with being able to store even small sets of plants on the same day. We were fortunate that the University of Nebraska-Lincoln hired Yu Han, who's a new assistant professor in the Department of Statistics here. And he did his PhD work at Iowa State, working with also phenotypes, but very different phenotypes. So he was looking at the gravitropic response in maize roots, and again was building these models that allowed him to both fill in for missing data and also provide more accurate estimations because you can share data from similar data points. And this work from his PhD just came out last year in actually I forget the journal name off the top of my head, but a statistics journal, not a plant phenotyping journal, unfortunately. So I was sitting down and talking to Johan at one point, and we said, okay, it would be good to adapt these methods to working with plant data, but we need to know, first of all, do they work? And for that, we need a useful data set that we can play around with. And fortunately, we had such a data set, which was generated several years ago, back when the greenhouse could only hold about 200 plants. So these were, all the plants were imaged every single day. This was a very early phenotyping paper that was published a couple of years ago with another of my collaborators, Yufeng Ge, who's a biosystems engineer. We had only two sets of factors, so we had a treatment, and the treatment was either drought, where the plant stopped being watered, so a pretty extreme drought, or not drought, and then two genotypes, either B73 or mini maize. And this is specifically fast flowering mini maize A, which was developed by Morgan McCaw at University of Missouri with Jim Birchler, and I want to make sure I thank him. We ran out of seed. It should be easy to bulk a fast flowering mini maize plant, but we were going through seed very quickly, so he was willing to share some additional seed with us. And at this very early experiment, we started imaging only 6 days after planting, so about when the plants emerged, and then imaged through 26 days. So this is actually a relative. I would say this does not play to our strengths at the institution, but in the early days we wanted data that was comparable to what was being generated using other facilities. So this was the data set we could use. And one of the things I want to emphasize on this slide is that phenomics data, it's like high throughput sequencing data, there's a lot of potential for reuse of data sets. So I was very happy that we had done a good job of annotating this data and storing it properly so that we were able to go back and reuse it for another purpose and not have to run another very expensive experiment. There are a number of data sets like this on the lab website. If anyone would like to use them for other purposes, you should feel free to. We want to encourage reuse as much as possible. So we can dig into this data set and look at the individual growth curves. So this is just plant area, total number of pixels. As I said, we're avoiding all of the interesting complicated phenotypes that we're starting to learn how to extract from image data. And you can see that there are obvious differences both between the two genotypes, the B73, which is the reference genotype for maize, and this fast flowering mini maize, which is a much smaller plant with a faster life cycle. You can also see that if you stop watering plants, they don't grow as fast, which is not a groundbreaking finding. But again, there's this significant difference. You can also see that this isn't entirely clean. The data for individual plants will bounce around a little bit from day to day. and there's a fair bit of overlap as we plot these individual plants. So what Yuhang's expertise is in this functional data analysis, where you fit a non-parametric curve to the data, which is very flexible in terms of having limited numbers of assumptions, and it allows us to calculate confidence bands around that estimated effect. It also allows us to separate out the effects. So we can plot for individual plants. We can also plot for groups and separate out the effects of different treatments or different genotypes. Now I'm going to click through these next couple of slides slowly, but I'm not going to say very much because I'm not a statistician. And I have to admit that I could not re-implement this model myself if I tried. The details of this are in the published paper, and if there's anything that is unclear in the paper, please do reach out to me or to Yuhang, and we can make it a bit more clear. So essentially we're fitting splines to the data and using cross-validation to figure out how much smoothing makes sense, how much is actually removing real signal. I'm not sure what to say about this slide, but I'm going to show it for a couple of seconds, and if, as I say, all of this is in the paper itself. But one of the things that does make sense to emphasize at this point is once we fitted these smoothed splines to the data, it allows us to both estimate what measurements should be throughout the entire course of development, but also to look at the first and second derivatives of the data, which is useful. So what you see here is simply the growth functions first for each of the 4 possible combinations of 2 genotypes and 2 treatments, and then the functions that were estimated for the effect size of both genotype and treatment. So the genotype effect is the predominant one early on, and then as the plants start running out of water and also getting larger, so their water demands get higher, the drought stress effect becomes larger. But as I highlighted in the previous slide, one of the really interesting or really useful things about this is that we can get the derivatives of these curves from the same math that gives us the curves themselves. So we can also look, I'm jumping ahead myself, we can also fit confidence bands around these effect sizes. So you can see that the effect of drought stress, we're relatively confident in the pattern. If you look at the genotype effect, the error around the genotype effect gets larger as we get later into the experiment, which is indicating that there's a lot more variance both within the B73 plants and within the fast-flowering mini-maize in terms of how large they are. And then this would be the slide with derivatives. So this is, again, the first derivatives of all possible pairwise combinations of factors and genotypes, and then of the effect sizes. So you can see that the genotype effect, the rate of change for genotype effect stays about the same throughout the experiment, whereas, as we could see from the previous curve, the effect of the drought stress accelerates throughout the experiment. So I always have to think back to high school physics, and so that we've got the position, which is the trait we're measuring, and then the first derivative, which is the velocity of change. I could also show you the graph of the second derivative, which would be acceleration. I'm still trying to wrap my head around what acceleration means in terms of a plant phenotype, but we do see that there's useful data in the second derivative. It's just hard to tie it back to something biologically meaningful so far. Right. So the reason we got into this is that we have this problem. We can't. We have a relatively small number of plants we can grow at any one time, and even if we use the whole greenhouse facility, we can't image all the plants on the same days. So using the same non-parametric functional regression, we can look at what our accuracy looks like if we look at all the plants, say, every second day as opposed to every day, and we do sacrifice a little bit of accuracy, say only about 1.5%, so from sampling every second day. So this tells us, in fact, that we probably could sample less, and I believe we took this down to once every fourth day, and we were still getting pretty good accuracy. And it did not work to go once every eighth day when you only have 20 data points, because that, I think, puts 2 points on the curve. But I think if we had a captured longer period of time, we would be able to do even more subsampling. The thing that really excited me about this, and I had to explain this to my statistical collaborators, because to them it was very obvious, is that instead of subsampling every second day, we can also subsample the data looking at some plants on one day, other plants on the second day, and back and forth like that. And again, we are sacrificing very small amounts of accuracy and measurements, which allows us to project the missing data points and be able to make direct comparisons. You'll also see that the error tends to be higher earlier in the experiment. Remember, this is a measurement of relative difference. And this is something we've seen in a number of different experiments that have been analyzed in my lab. There's a bit more about this. This is another paper that came out last year by a grad student of mine, Jikai Liang. What he found is that if you look at just the heritability of measurements, so the proportion of total variance that is explained by genotype, that this heritability goes up over time. Excuse me. And so what we think is happening here is essentially the amount of error in measurements from measuring a plant using image data is relatively fixed. So as plants get bigger, your absolute values are larger, so the error as percent of the value is going down. And once you subtract out as the error gets smaller, more of the remaining variance is left over to be explained by genotype. So again, this is nice, because this is what we're set up to do, is work with really big plants. So now for something slightly different. As I said, we're set up to work with really big plants, but really big relative to seedlings. So there's such a thing. There are situations where hybrid bit vigor is a bad thing. This is a photo from another experiment we ran working with some recently off-patent maize hybrids. What will jump out to those of you in the audience who are familiar with corn is that these plants coming through the imaging system don't have tassels anymore. We were able to grow the inbreds perfectly well. The hybrids had a little bit too much vigor. They were over the maximum height, so we had to unexpectedly curtail the experiment when the tassels started hitting the walls as the plants were coming through the system. So that clearly was not a strategy that was going to give us interesting data sets to play around with. Fortunately, we have this close relative of maize, which is also a substantial crop both in Nebraska and around the U.S., which is sorghum. A lot like corn in a lot of ways, more drought tolerant, smaller genome, much harder to cross, and most importantly, the material that we grow in the U.S. is usually substantially shorter than corn. Now, there are still tall sorghums. Here you see a couple of folks in my lab collecting some field data from some cuts. So there's enough genetic diversity even if we look at stuff that is short enough that we can grow it in the greenhouse. So by this point, we'd learned a lot of lessons. This experiment was run over the last year and is in fact still ongoing. So we selected from an existing association panel, the CASA Sorghum Association panel, about 400 lines. We picked about 300 of those based on field phenotyping to select lines that would be less than 2.3 meters tall at flowering in the greenhouse, so we would be able to grow the plants without having to chop off the heads, which is very depressing and not particularly agronomically relevant. This panel had been grown for 2 years in phenotype here in Nebraska, and there are also many other published phenotypic data sets generated for the CASA Sorghum Association panel. Another lesson that I have learned in the couple of years I've been doing this now is life is so much easier if you can work with genetic material where someone else is also doing the hard work of generating ground truth data for you. Otherwise, actually even so, you spend probably a lot more time just making measurements of plants than you would if you weren't trying to do high-throughput phenotyping in the first place. We'd also learned that the small seedlings, there just isn't a lot to see on a high-throughput phenotyping system, and the error tends to be a lot larger. So these plants were grown off belt for about a month and a half and then imaged for about a month, and we tried as much as possible to include flowering time in that window. The majority of these lines already had about 400,000 SNP markers, which had been genotyped for them. And then we did an additional round of genotyping with 150,000 SNP markers across all the lines that we had grown. And this was and is really exciting because we finally have a data set large enough to map back to causal SNPs or causal regions of the genome. And given my own training and predilection for wanting to understand, what gene does what, that made starts to make life a lot more fun. We also collaborated. Brandy Sigmund, who's a research assistant professor here at University of Nebraska and has been the person growing these in the field. So she has both been phenotyping in the field and then phenotyping the same plants that come off of the greenhouse system. With an interest primarily in inflorescence architecture, and we're hoping ultimately to be able to derive a lot of these same traits from the image data. But when you look at these well-defined traits that are sort of terminal traits that are taken at the end of the growing season, we're able to do very good mapping and identify relatively small numbers of high confidence candidate genes. And that's how conventional quantitative genetics works. The problem we're running into, and the problem that this talk is focused on, is how do we deal with time series phenotypes? So this is simply plant height collected from that imaging data, and how do I describe this as a trait? I mean, I could take the height at the end of the experiment, but clearly we're throwing away a lot of data about the shape of the curve and when the plant grows. You can also see that this shape would not fit we wouldn't be able to fit a single logistic curve to this data. And of course, we can also look at these first and second derivatives. So the first thing to do is just throw computational resources at the problem. Unfortunately, I have grad students who are happy to do that. So what you see here are separate GWASs for plant height on every single day when image data was collected. You can see that the days after planting is ticking up in units of 2 because plants were only imaged every second day. And what was striking to me when we got this analysis was, first of all, we don't see a lot of genes controlling height. Normally in the Sorghum Association panel, you are able to map all of them. It sort of made sense that you wouldn't be able to in this case just because we've thrown away a lot of the really tall lines, which would be the ones that have wild-type alleles of dwarfing genes. But the other interesting thing is that we are able to pick up dwarf 3, which is on chromosome 7, early in the experiment, and then that fades away entirely as we're going a little further along. And I was wondering, so why are we losing the signal for later in development? And we can look at the curves for the individual plants, and the data looks really, really messy. Let's see. But this, I think, is what's happening. So what you see here is a single sorghum plant over a period of about 2.5 weeks. And well, that is a really noisy GIF. Anyway, what you are seeing here is the actual emergence of the panicle. And if we, instead of looking at the days after planting for each individual genotypes, we line them up by when we first observe the emergence of the panicle. We can start to see much cleaner patterns emerging in the data. Some genotypes you see this large increase in height around the panicle because you have substantial head exertion. In other genotypes, you really don't see that because the panicle stays down in the leaves. So we took the exact same height data, and instead of comparing the same number of days after planting for every single genotype, we instead looked at the number of days before or after the panicle could first be observed in the image data. Oh yeah, just messy data. And now you see this is another version of the same GWAS plot. We have both dwarf 2, dwarf 4, and dwarf 3 can be picked up at various points. Dwarf 1 almost comes to statistical significance. And we also pick up a couple of additional loci that show substantial significance. I think those additional loci are probably not actually unknowns. They're probably known flowering time genes. And Shenyang is doing that right now. The other much more embarrassingly basic problem that you run into with this type of data is I can show you these animated graphs, and which are hopefully coming through the WebEx all right. But it's very hard to look at this and really think about patterns other than to see, okay, look, I can identify some things at some time points and other things at other time points. Oh, thank you, Carolyn. So we need another way to visualize this, and this was stolen completely from, I think, a paper by Edgar Spalding that was looking at QTLs. So what you see here is on the y-axis, we have positions along the 10 chromosomes of sorghum, not labeled yet because we're still implementing this in matplotlib and Python. And then on the x-axis, we see each of the days after planting. Each column is a separate GWAS, and then we're looking at windows across the genome so that we actually had something we could plot on here. So in the days after planting analysis, we really only get one substantial, which is that dwarf3 gene, and we lose that the signal from that gene after about 10 days into the experiment. If instead we do this, we line plants up based on when we see the panicle instead of when we planted them, we're able to pick up many more statistically significant loci across multiple days. So the signal from dwarf3 actually is not as strong anymore, but we are able to pick up dwarf2 and 4. As I said, dwarf1 doesn't really show up a lot, as well as 3 other loci that rise above the threshold of statistical significance at some point or another. All right, so yes, I think I already gave this disclaimer, which is that I think the unknowns are probably actually known maturity loci. And there should be a second disclaimer here. Oh yes, this is based on only a single replicate of the panel, so I would never try and publish this. The second replicate is being analyzed as we speak, and the third is actually growing in the greenhouse right now. And we'll start collecting data in a couple of weeks, I think. So once we have 3 of these, we should actually be able to say something with confidence. So the first key message from this, from what I've shared so far, is that I think having both the equipment and the people to image full experiments all at one time is really one of the primary bottlenecks in being able to do large-scale quantitative genetic and/or breeding experiments right now using high-throughput phenotyping. And using techniques like functional data analysis that don't require making assumptions about how traits change over time, but can infer missing data points, at least for things that show trends over multiple days, means we can do 2 to 4 times the size of the experiment for essentially the same cost, and makes it more feasible to actually be able to identify interesting genes or new varieties that are better adapted to new climates. Any of the stuff that is probably the reasons we got into either biology or breeding or high-throughput phenotyping or what have you. So the data I've shown you is from a greenhouse study. I'm hoping to soon have data to test whether this really does work in a field setting. We're doing that in 2 ways. One is this cable-based system that the University of Nebraska has invested in. We have about a 1-acre footprint with 360 separate 2-row plots under it. We're going to be able to move this payload system back and forth over it, collecting both image and thermal data throughout the season, as well as being able to treat with different water levels. And then secondly, working with more conventional drone-based systems with my colleague Oscar Rodriguez, our popcorn breeder, and Yayin Shi, who's another engineer who's focused more in the unmanned aerial systems, and I believe came out of Texas A&M, and I think actually worked with Seth Murray, one of our hosts, at one point. So we were fortunate to be able to grab her up here at UNL. The second key message, and I think maybe this one has broader applicability, is that the assumption of things like QTL mapping or GWAS is that we're comparing the same thing between different genotypes. And that's relatively easy at the seedling stage, and it's relatively easy for terminal stage phenotypes. In the middle of the plant growth cycle, though, I think it's a really hard thing. Plants develop at different rates. You can look at variation in total life cycle. Within that life cycle, different parts will be shorter or longer. So how do we actually make these comparisons. So in the short term, I think we're developing much better techniques to be able to pick which data points are going to be the most equivalent between different genotypes, different treatments, and different environments. In the longer term, there are statistical techniques for actually comparing the shapes of curves over time rather than having to do point-wise comparisons. This is relatively straightforward for parametric models because, again, the shape of your curve is defined by a relatively small. So I did a rotation years ago in a circadian biology lab, and that was so nice, right? So essentially every pattern of circadian regulation you come across is defined by only 3 parameters: the phase, the period, and the amplitude. And so everything can be reduced to those 3 factors. However, there are techniques for being able to compare the functions and curves derived from non-parametric models that statisticians know about. It's just a question of being able to find people with expertise and pull them into plant phenotyping and get them excited about these problems, which means we need fun data sets, fun and well-annotated data sets for them to play with. And at the very least, if you're doing anything that is looking at the middle part of plant growth and not seedling stage and not mature trait, it really makes sense to control for flowering time, which is not a new message by any means here. That was a missing line there. So I mentioned at the beginning of the talk, and the sort of work I'm talking about here is not something that my own lab could do on its own. We don't have the expertise. So we're very fortunate to work with some great engineers. I mentioned Yang Shi. A lot of this data was generated out of Yufeng Ge, who's I think I mentioned an engineer here, and a couple of members of his lab, including Piyush and Frank. Frank we've just hired to manage a phenotyping facility, so we're going to get to hold on to him long term. Similarly, the statisticians who we've been willing to play around with phenotyping data sets that none of this would have been possible. Yu Hong, who was the lead author on the paper that inspired this presentation. Yumo, who's also an author on there and is just a really great collaborator. And Jennifer Clark, who runs the Quantitative Life Sciences Institute out here. And then, of course, also the plants and computer bits. So I'm very fortunate to both the expertise I have in my lab. A lot of the work from members of my lab that I've shown it comes primarily from Chenyong Miao, who's a second-year PhD student, and Jikai, who's a third-year PhD student, and then our collaborator, Brandy Sigmund, who's a real geneticist and field biologist. It's also, fortunate that this phenotyping work gets supported by a lot of diverse sources, which is good because it's both expensive and labor-intensive. And I also really want to thank the Plant Phenome Journal and the organizers both for accepting the paper and for this opportunity to present. It's a real challenge figuring out where to send papers that are more about, hey, we had this interesting problem and this is how we solved it, but don't make that next step to the biology. One solution would be just to wait until we've solved an interesting biological question with our solution, but I think that slows things down. I mean, it can slow things down by several years sometimes. So it's nice to have this forum to be able to put out, here's the solution, and hopefully it's useful to other people before we've actually answered our biological problem. And with that, thank you all very much for your attention.
Thank you, James. I always feel like at the ends of these things we need to clap because there are so many people that are online and you can't even hear it. So, yay, thank you. So we have a good deal of time here, about 20 minutes, for question and answer. I'll open it up first, let's see, to anyone that is on the call that's not me.
Oh, I've got something from Seth already. Okay. So Seth, I guess I can repeat hopefully everyone can see the text chat, but if not, Seth is asking if terminal phenotypes cannot be anticipated from early growth, as you show. What does this say about the utility of using automated greenhouses that only do seedling screens? Mm-hmm. I think it depends a lot on what traits you're looking at. I mean, a lot are, more at the level of photosynthetic efficiency or very basic physiological traits. It's probably more likely that you can get useful predictions from seedling or relatively small phenotypes. If you're looking at things like plant architecture, resource use efficiency, yield, I don't think that doing seedling screens is the right way to get at those.
So I have a different question. It's sort of at a different level. It's not really about the biology.
Okay.
The work that you do, you involve expertise in really diverse areas. How do you build those teams? How do you bring in people when you know you have a gap, and how do you make it worth their while? Because this kind of bringing teams together and working through it well, it's not easy.
No, it certainly is not. So I can say a little bit about what I've done, but I think it's really important before that to say, I mean, one major advantage I have has nothing, I can't take any credit for, which is that I was hired at the University of Nebraska at a time when our administration had decided to do a major renewal of faculty. So a lot of people had gotten buyouts, and a lot of new professors were hired. So it was like the first week in a new high school where no one knows what table to sit at. It was a lot easier to build these collaborations because nobody knew what they were doing yet. And if you look at, I think Oscar is the exception. Almost all the faculty I'm working with are people who were hired in the last 4 years. And that's not anything intentional. I think it's just that those were the people who had sort of for collaboration. So that's the thing. I was good fortune that had nothing to do with anything I did myself.
That was just people with bandwidth available.
Yeah, good people with bandwidth who are available. In terms of how you sustain those, though, I think that important thing is you have to understand everyone's incentive structure, and that can be very different for a computer scientist or an engineer or a breeder. And then be willing to always feel like, another question I'll jump to in a second. You're always going. It's like any relationship. You're always going to feel like you're doing more than your share, and
right.
You recognize that the other people do as well.
Right.
And then finally, being really good about giving credit where credit is due. I tried to have everyone through this talk, and whenever I go anywhere, I try and explain. These are really good folks. Usually that helps a lot. Occasionally it backfires when your collaborators get poached away by other universities. But what can you do? All right. Katie or Caddy, you've mentioned a few times that you collected field data as well as greenhouse. I understand you still need to ground truth it, but can you make a preliminary comment on the correlations you have observed between field and greenhouse? So for some things, the correlation is quite good. So if we look at height or flowering time for plants grown in the greenhouse in the summer, we see very good predictions between the field collected data and the greenhouse collected data. For a lot of other things, we see very good correlations. Again, more complex traits, water use efficiency, biomass yield itself. A lot of things that have more to do with biochemistry, I'm optimistic we'll see better predictions than the traits that are, I guess, closer to fitness, that are the output of so many different factors in the plant that all have to work together to determine something like biomass yield.
Okay, are there other questions? Give it a pause. Oh, got one. Two.
Oh, all right. Let's see. Let's start with Matt. You mentioned that you can phenotype subsets of the plants on different days to overcome the plant number limitation. Are you replicating the plants on those days to have complete blocks and incomplete blocks for the unreplicated genotypes, or how are you controlling the day effect for phenotyping? So in the data that I have shown, there is one check genotype that is replicated across both houses. But other than that, the GWAS results I showed you, that is one plant per genotype with only one replicate. So we really are constrained by the number of plants we can grow in a greenhouse at a single time. This is something a lot of the folks I collaborate with a lot of the folks who run the facility, I think their training is in physiology. So I think they think that I'm very unreasonable, because for them, if you have 40 or 50 plants, you can do an interesting experiment. I need hundreds of genotypes in order to be able to say anything. So it would be experimental design in the greenhouse matters a lot. We definitely see row and column effects, and when we can, we try and correct for that with check genotypes. We don't have complete replicated blocks in the same grow out. I would love to if I had space. All right, Steve. Do you think implementing 3-parameter logistical models are appropriate when you are only phenotyping, say, the first 40 days, as terminal height has not been achieved and the maximum growth rate may not be achieved yet? How do non-parametric models address this question, as it seems they are somewhat assuming a terminal plateau when the model is plotted similar to logistic models? So yeah, there are two different problems. So one is interpolating missing data. The other is projecting out beyond the range of data that you've collected. And so I don't think that non-parametric models work very well for projection. They work really well for interpolation. So I've got data throughout this growth period, but I'm missing data points, or maybe there was a thunderstorm, so we didn't fly for a week, didn't fly the drones for a week or something. For what you're talking about, where you've collected part of the season and you're making projections farther out, you really do need to make a lot of assumptions about how the plant is going to develop. And since you need to make those assumptions anyway, I would say logistical models are going to work much better for projecting outside of the range of what's been observed than non-parametric models.
Very nice. You've got another question.
Okay. Acer, so you see interesting snips pop out of the GWAS at different time points. Do you still pick these up when you were looking at the functions? In other words, do you miss important variation when you model traits as functions? So for the non-parametric functions, and we haven't tried parametric functions in this data set, we've run the GWAS either with the raw data or with the values of the functions at the time points when we collected the data. And the results are about the same. If anything, they're a little bit better when we look at the point on the curve of the function rather than the value itself. I think that's because we're borrowing data from adjacent time points. So the curve may actually be a more accurate measurement of the true value of the trait at that time than the measurement of the trait from the image.
Very nice. Thank you. Okay, anyone else? While we still have James on the hot seat here. Okay, well, with that, let's thank James again. And I'll put in one last plug for the Plant Phenome Journal. If this is the kind of work that you're doing, this is where it belongs. When you're marrying the methods that you're generating with this general area of plant phenomics, we'd like to see your paper. So we encourage you to talk with one of the editors and to submit your manuscript. Thank you again, James.
Thank you.
Thanks to all of you for coming. Bye-bye.